All right. So today is going to kind of act as an introduction to cell respiration, because that's our big, one of our big ideas for this unit. Not like one of the College Board big ideas, one of our in-class big ideas. So where's my clicker? There it is. Cellular energetics, the name of this unit, applies to cell respiration, fermentation, and photosynthesis. Welcome. We just got started. We are using a basic outline for cell respiration and fermentation, and this is that basic outline, which is catalysis or breakdown. When we are using cellular respiration, we are breaking down sugars for energy, right? And we do that through a redox reaction. Okay. How many of you remember learning about redox reactions but might not have that vocab down? Nobody. Okay, cool. Not sure where chemistry is in relation to bio. It can get weird sometimes. So, redox reactions. That's our big idea for how we get our overview of cell respiration. My ideal outcome for this lesson, whether that be after break or after class today, is for you to be able to describe what a redox reaction is, and then tell me why cell respiration uses redox reactions. That's my goal. So, to start us off, we got a little bit of a review about catabolic pathways. Remember, we had catabolic pathways and anabolic pathways. Catabolic pathways are breakdown pathways. Remember, cats like to push things off of tables. They like to break things. Catabolic is breakdown. What is happening is we are oxidizing organic fuels and releasing energy. That's what chemically happens in cell respiration. We are oxidizing organic fuels. When we do this, we are transferring electrons from these fuels to other molecules. And that process is essentially a majority of what cell respiration is. Or it plays a super big role in what cell respiration is that transfer of electrons from these fuels to other molecules in the process of cell respiration. So our little diagram, we've got our big organic molecule. Most of the time these are carbohydrates. They can also be lipids and all that fun stuff. Most of the time it starts with a carbohydrate. We break it down into its pieces, and when we do break it down into its pieces, we get energy. That is a catabolic reaction. And a big step in catalysis is this oxidation. That's the ox in redox, which we'll get to in 2 seconds. But it's all about electrons. Bless you. All about electrons. We're going to talk a heck of a lot about electrons. Yes. Does it? Brain broke. It's supposed to say electrons. Believe it or not, chemistry, not very democratic. There we go. No, don't apologize. That means I can use it again. It be not that. All right. If you haven't noticed, there's a trend of this in this class of me apparently not knowing how to make a proper PowerPoint. So, one of those skills you learn over time, I guess. So, catabolic pathways, breakdown pathways. We had the reverse of this with anabolic pathways. Anabolic pathways are build-up pathways. Catabolic pathways are breakdown pathways. So, this is how we get to our fermentation, aerobic respiration. This whole process, or these two processes, rely on these catabolic pathways. These are catabolic pathways. Fermentation is where we partially degrade sugars without oxygen. Fermentation happens without oxygen. And we'll get to this whenever we talk about fermentation more in depth. But when we are running or doing any other physical exercise and our muscles don't get enough oxygen because we're using up oxygen faster than we can breathe it in, our muscles turn to fermentation because we don't have oxygen. And that fermentation creates lactic acid, and that is why our muscles hurt when we do exercise. But we'll get to that when we talk about fermentation. We'll have like a whole couple days with fermentation. Aerobic respiration happens when oxygen is consumed as well. This aerobic respiration is what we do on a regular basis. When oxygen is not used, it is called anaerobic respiration. Anaerobic respiration. Make sure I keep those straight. Most eukaryotes and many prokaryotes do this aerobic respiration, use oxygen. Not all of them, though. Some of them have pathways that don't involve oxygen at all. Some of them have backup pathways. So when oxygen's not around, they can still, do metabolism. But it's not their preferred method, and that is called anaerobic respiration. So when a process uses oxygen, it's called aerobic, and when a process does not use oxygen, it's called anaerobic. So when we've got fermentation, we're going to degrade stuff without oxygen. Fermentation is a type of anaerobic respiration. So if we did a mini concept map, because I love a concept map, if we did a mini concept map, we can have respiration broken down into two categories, aerobic and anaerobic. And we can take anaerobic and have a little offshoot, and that is where fermentation pops in. So if that helps organize thoughts, I love a concept map. So you don't have to have it down, obviously, but it helps me. So many prokaryotes do aerobic respiration along with us, most of us eukaryotes do aerobic respiration just because it's more efficient. Oxygen's really good at what it does. Yeah, that is the baseline, what we are going to talk about, what these processes are. These are our big equations, our big processes for cellular energetics. So, not completely done with the chemistry, but moving a little bit closer into bio than we were earlier this week. Questions here? All right. So, what is cell respiration specifically? So we've talked about cellular respiration. I've mentioned it a lot. Cellular respiration is not just aerobic. Cell respiration is anaerobic and aerobic processes. I flipped anaerobic and aerobic again. Cell respiration has multiple steps. Some of them are aerobic, some of them are anaerobic. Technically, and scientifically, cellular respiration is the sum of all of those aerobic and anaerobic processes that a cell uses to break down fuels for energy. But a lot of the time, when people are talking about cell respiration colloquially or just out and about, they are referring to only the aerobic processes. But it's actually both. So make sure whenever you are using cellular respiration, you are paying attention to whether or not the process you are talking about is aerobic, anaerobic, or both. This is just a quick summation of the cellular respiration process in the mitochondria. We've got some steps that do not require oxygen and some steps that do require oxygen. So the whole thing is cell respiration. It's not just the parts that require oxygen. Cool? Little disclaimer. All right. So, when we talk about aerobic respiration and cellular respiration, we've got like aerobic respiration is essentially like an overarching term. It is a type of respiration. And this is our like skeleton equation for aerobic respiration. We've got some organic compounds with oxygen, which yields carbon dioxide, water, and energy. That's our basic skeleton equation for aerobic respiration. Cellular respiration has a specific equation, and you are going to use this so much you'll have it memorized. Also, because photosynthesis is this equation backwards. So if you've got one, you've got them both. So cell respiration is C6H12O6. That is glucose. I can find my pens. Yes, please. C6H12O6. That right there is glucose. Nope. Six O2 molecules. That is 6 oxygens. Oxygen likes to hang out in twos. I'm just happier that way. So, a glucose molecule and 6 oxygen molecules will be transformed into 6 carbon dioxides, 6 CO2s, 6 water molecules, 6 H2O, and energy. That energy is preferably in the form of ATP, but sometimes it is lost as heat in the transfer. So it's not a perfect process, but it gets the job done. So our cellular respiration equation is glucose plus oxygen is carbon dioxide, water, and energy. So C6H12O6 plus 6O2 gives us, went backwards. 6 CO2 plus 6 H2O plus energy. That is our equation for cell respiration. That is what our next couple weeks are going to be over. I believe it ends in an escape room, so that should be fun. And then photosynthesis ends in a lab. So just have that in mind, what we're working for. All right. So how do these pathways yield energy? How do we get from the decomposition of glucose to energy? It is these 3 steps. We're going to transfer electrons. Change it to elections again. That one's not on me. That one's on spell check. We're going to transfer electrons. We're going to release that energy that was stored in those compounds, and we're going to make ATP. Those are our 3 steps. Now, we're going to learn about those 3 steps in a level of detail you haven't quite gotten in biology before, but that's okay. That's why we're here. That's why it's AP biology. This is how we get from glucose to energy. We're going to transfer a bunch of electrons, we're going to release some energy stored in organic compounds, and we're going to make ATP. That's the goal. That's the money. Ooh. That's not good. All right. It's not broken. All right. So those are our 3 steps. So how we get to these 3 steps is through a redox reaction. Redox reaction. Synthesize ATP. Redox reactions are a 2-step process, or 2 big things that happen. That is oxidation and reduction, or reduction and oxidation. Redox. Reduction, oxidation, redox reactions. Oxidation is the loss of electrons, and reduction is the gain of electrons. So when we are transferring an electron from one thing to another, like we said was our big step here, transfer of electrons. When we are transferring electrons, one material is going to lose them, and the other material is going to gain them. So that's why we call it a redox reaction, because we are one molecule is going to lose an electron, and another molecule is going to gain an electron. So we call it a redox reaction. This happens when one or more electrons is transferred from one reactant to another. Oxidation is losing, reduction is gaining. It's kind of counterintuitive because reduction kind of colloquially means get smaller, but it is actually gaining electrons. It's called reduction because the charge is being reduced, because electrons have a negative charge. When we are adding a negative to something, it's going to be reduced. So oxidation is the loss of an electron. Reduction is the gain of electron. We are reducing its charge because that negative electron. It's kind of something you got to look at upside down and backwards to make it make sense, but it can make sense. Oxidation is the loss of an electron. Reduction is the gain of an electron. So when we are transferring from one material to another, one's going to lose, one's going to gain. It's going to be a oxidation, reduction, redox reaction. Cool. All right, we're going to go over this 9 ways to Sunday. So here is our skeleton equation. So like our bunch of variables, nothing specific equation for a redox reaction. We've got reactant A, reactant B. We are going to change that electron. We're going to transfer that electron from A to B. A is going to lose an electron, so it is oxidized. And B is going to gain an electron, so it is reduced. We're going to transfer that electron. So now A has a positive charge because we took a negative away. And B has a negative charge because we gained that electron. That means A is going to be oxidized. And B is going to be reduced. So, this equation down here. What happens to iron or iron-containing metals if you leave them outside in the open, in the rain, and the snow, and the wind for long periods of time? They rust. You know what rust is? It's oxidation. So, right here, we've got iron and oxygen. When iron is exposed to oxygen, that iron is oxidized and that oxygen is reduced. So this is a redox reaction we see all the time. And we try really hard to keep from happening on our cars. Well, if you're me and you're used to drive old cars. Oxidation, reduction. Something's going to lose, something's going to gain. Questions here. There's a lot of weird backwards terminology that comes up with this stuff. So, oxidation, reduction. So, we have another word to add to our repertoire. When our electron donor is going to be called the reducing agent, and our electron acceptor is going to be our oxidizing agent. So, our electron donor, our A in this example, because it's donating its electron, this is the reducing agent. And this B from our example, it's going to gain an electron, so it is our oxidizing agent. Our reducing agent is oxidized, and our oxidizing agent is reduced. So, we've got another little example right here. Our reducing agent is oxidized, and our oxidizing agent is reduced. This diagram right here is probably going to be the most helpful for you. So, if you want to do it together, we can do it together. I don't mind. So, we'll keep the colors semi-straight. So, we start with A and B. A is going to be our reducing agent. And B is going to be our oxidizing agent. We are going to move electrons from A to B. So, A is going to lose electrons, bless you, and you will see electrons abbreviated as E minus. B is going to gain electrons. So our reducing agent is oxidized. And our oxidizing agent is reduced. This whole thing is a redox reaction. Questions here? Doing okay? All right. So, cellular respiration is a redox reaction. So if we have our equation, glucose plus oxygen gives us carbon dioxide and water. Glucose is going to lose electrons, and oxygen is going to gain electrons. So if glucose is oxidized, if it's losing electrons, that means glucose is our reducing agent. And oxygen is going to gain electrons, so it is going to be our oxidizing agent. The only thing that's different here than with our skeleton equation is that hydrogen is going to move with those electrons. That's where the hydrogens come into play. The only thing here is that wherever those electrons go, hydrogens are going to follow because it's easier to transfer them that way with those hydrogens. So, hydrogen is transferred from glucose to oxygen. That's what this main equation tells us, right? But our electron changes and the hydrogen is transferred to oxygen. This equation doesn't show us that electron change. It just shows us where the hydrogens are changing. But if we want to understand this on an energy level, we have to understand that with those hydrogens come electrons. With those hydrogens come electrons. So our glucose is oxidized. It's going to transfer some electrons to oxygen. Because remember that redox reaction is going to happen between those two reactants. But the hydrogens are going to follow. Because our hydrogens are going to go from glucose. Because remember, our glucose is C6H12O6. And we have 6 C's and 6 O's, but those 12 hydrogens got to go somewhere, and they're going to go to the water, which is that oxygen. So we can kind of trace where those electrons are based on where the hydrogens are. Doing okay? All right. Some fancy orgo today. All right. So, where does this energy come from? We've talked about electrons. We've talked about releasing energy, right? That was step 2. Energy is released from those organic compounds. Where on earth is this energy coming from? When we are oxidizing glucose, when we're transferring those electrons, we are moving them from a high energy state to a low energy state. We don't necessarily, in this class, in this course, need to know what it means to be a high energy electron and a low energy electron. We just need to know that we can go from a high energy state to a low energy state, and that transfer releases energy by synthesizing ATP. That's how we get our energy. So, the energy state of the electrons in our glucose is going to be way higher than our energy state of our electrons in our water or on that oxygen. And when we transfer those electrons from that high to low energy state, in something called the electron transport chain, that's when we get to synthesize ATP. That's when we get to make that energy. Or make what we are going, we're not making energy, but we are making something that can store that energy. So, energy harvest. Again, it is this 3 steps. I know it looks a little wonky. First things first, glucose is broken down in a series of steps. We're going to talk about them next week, or not next week, but the week we get back. We're going to talk about glycolysis and the Krebs cycle. Calvin is first. That's our series of steps. When we are breaking down glucose, we're going to strip electrons off of there. We're going to yank off those electrons as we go. But we have to have a place to put the electrons. And where we put those electrons is an electron carrier. The electron carrier we use is called nicotinamide adenine dinucleotide. You do not need to know that big long word. All you need to know is NAD. It stands for nicotinamide adenine dinucleotide, and it's fun to say. But we're going to take those electrons, and we're going to take those hydrogens. Yeah. Via. You're good. It's me. I saw that one and meant to go back and change it. I didn't. That one is on me. Nope. Via. Don't know what it is. We got it. Hydrogen is going to be passed to these electron carriers, specifically NAD. And we're going to do that with hydrogen. So we're going to take some of those hydrogens, take those electrons off of there, transfer them to a carrier. That carrier, NAD, is really good at what it does. And it's really good at what it does because it is very, very easy to switch from its oxidized form to its reduced form. Its oxidized form is NAD+, and its reduced form is NADH. So NADH has the hydrogen, which means it has the energy. It has the electrons. NAD+ is oxidized. It's going to lose those electrons. So this is how our electrons get off of oxygen or off of glucose. We've taken those electrons off of glucose and we put them on a carrier. But now that they're on a carrier, we still need to synthesize ATP. We need to get from our carrier to our end product. So we've taken glucose, we've broken it down, we've oxidized it by taking those electrons. And when we take those electrons, we're going to put them onto our carrier, onto NADH, or onto NAD to make it NADH. NADH is going to carry those high energy electrons until it can get to where it needs to go. Doing okay? All right. So, how does NAD trap electrons? How do we get those electrons from glucose to that carrier? A fancy enzyme called a dehydrogenase. Dehydrogenase removes electron atoms, or not electron atoms, hydrogen atoms, and electrons, and a phosphorus, to NAD. We're going to make it NADH and H+. And then we are going to use that NADH to make ATP. So an enzyme called dehydrogenase is going to take those electrons from those glucose decomposition molecules. They're going to transfer them to NAD+, and they're going to make NADH and some extra hydrogen ions. And that energy is going to be stored in NADH. That NADH can then be used to make ATP. So this is a little derivative of this. It's not in our description of cell respiration. We've got malate. It's a carbohydrate. Malate and NAD+ will meet something called malate dehydrogenase. You don't have to have this equation down, but it's just my example. Malate dehydrogenase will take some hydrogens off of this. You can see we lost a hydrogen here. And it will transfer it to our NAD+, and we'll have an extra one here. So malate dehydrogenase takes off hydrogens. It dehydrogens things. And it's going to transfer them to our NAD+. We're going to get NADH. Cool. All right. So now we can get those electrons off of those carriers and get them to oxygen. And we're going to do that via the electron transport chain. We're going to spend an entire day talking about the electron transport chain when we get back. Essentially, the electron transport chain is a bunch of proteins. There are a few other molecules in there, but mainly proteins that are built into that inner membrane of the mitochondria. And when those electrons are removed from glucose and on NADH, they're going to start at the top of this chain, the first high energy protein, and it's going to be transferred to a lower energy protein, to a lower energy protein, to a lower energy, until eventually oxygen is at the very end. Because oxygen is very, very good at taking in electrons, even when they're low energy. That's the whole point of oxygen. We need oxygen to breathe because it's very, very good at receiving low energy electrons, which we need in order to synthesize ATP. So oxygen is the last step in the electron transport chain. It's the last electron acceptor in our electron transport chain. So, questions here about how we got from glucose to oxygen. So this electron transport chain, this step in cellular respiration, happens in the mitochondria because those electron transport chain proteins are literally embedded in that inner membrane of the mitochondria. Remember that mitochondria has two membranes. All right. This is some fancy technical stuff. Okay, so this is our electron transport chain. How do we get from oxygen to water? When oxygen accepts those final electrons, because it's that final electron acceptor, those electrons come with those hydrogens. So right here, we've got a diagram of the electron transport chain. You don't need to have this one down because we're going to draw it in major detail later on. But I just wanted to show you. We've got our NADH with our electrons. Those electrons are going to get transferred to a protein in the electron transport chain. And when they do, we're going to release NAD plus and H plus, or those hydrogen ions. Those hydrogen ions are going to move to the very end, and when those electrons move down that chain, down that little ladder from high energy to low energy, we're going to synthesize ATP, and those electrons, along with those hydrogens, are going to join in with oxygen, at the end, and we're going to get water. We all right? All right. That's cellular respiration. That's the specifically the aerobic process because we've got oxygen. The Monday we get back from break, we're going to talk about glycolysis, which is an anaerobic process. But that is what I have for you today. Does anybody have any questions? I can go back if you need me to go back to anything. But yeah, that's cellular respiration in a nutshell. So, if you have any questions about these redox reactions, we'll practice them when we get back. These aren't gonna go away. Um, yeah, that's what I got. Yeah. Yep. This one or this one?